So we're moving into chapter 4 of section 1, getting away from first semester chemistry and into organic chemistry, which in most biology programs is the course that's the weeding out course. Is that pretty much any university that I'm familiar with, you got to take organic chemistry your second year, and like a Roach Motel, many students go in, not so many students come out. It is the way that they weed out the students that are going to be successful going on, in their opinion, from the students that won't be successful going on. So it is purposely incredibly difficult, and took me 2 tries to get through it. It's partly because of metacognition issues, which I talk about in the metacognition lab, but it's a tough course. It's a really tough course. This is another thing about going to school as a science major, although you'll run into it in other fields as well. They're in the bookstore. She goes, that'll be $100. He goes, $100 for one textbook? But it's required. If I don't buy it, I'll fail the class. Huh. You want it or not? Well, what if I get a used copy? That is a used copy. What if we only get the pages we actually need? Advanced science textbooks, $100 is, even for a used copy, $100 cheap. Is it $300-400? Not unusual. There was a very short period of time where they said to professors, for these books, you can pick the chapters that you want. We'll bind just those chapters together and it'll be cheaper. Apparently it was too cheap for them because they weren't making enough profits, because they stopped doing that pretty quickly. But you used to be able to just order the chapters that you need, which is usually less than half of the chapters in any given textbook. But at least you're not paying for a textbook here. And all the chapters that we have are all the chapters we're going to use. For people that are choosing to do an abstract with a fairly technical source, there's going to be a lot of authors, and you got to write them all down. And this is the author list, giving credit where credit is due. The first author, senior graduate student on the project, made the figures. The second author, graduate student in the lab that has nothing to do with this project, but it was included because he or she hung around the group meetings, usually for the food. The third author, first year student who actually did the experiments, performed the analysis, and wrote the whole paper. Thinks being third author is fair. The middle authors, author names nobody really reads, reserved for undergrads and technical staff. The second to last author, ambitious assistant professor or postdoc who instigated the paper. Kind of pushed everybody to do the work, didn't actually do the work themselves. The last author, the head honcho, hasn't even read the paper, but hey, he or she got the funding and their famous name will get the paper accepted. So when you see a lot of names, there are reasons why those names are there. And sometimes they put the whole name down. Here they're just doing the name in the initials. But when you do an abstract, if you use one of these sorts of papers, you have to write them all out. If you look at our reference list for the textbook, it follows the pattern that I want you to do for the abstracts in terms of title. And sometimes there are many authors listed. I agree with you, it's a pain in the butt. Okay, I already have a bunch of this stuff done. I'm going to black it out for a while. This was a bad morning in terms of me around computer systems. My computer that I left doing an update yesterday was still doing it when I came in this morning, and then didn't want to stop doing it, and then gave me a blank screen for a while. I literally had to flip it over, pull the battery out, exhaust the power with the power button, and then get it all to reboot. And even then, had to do it a couple of times. And the video system, for some reason, 1 minute and 46 seconds into this chapter, went, I'm done. And that's all I had. So I had to start it all over again. I was almost to the end of the chapter by the time I realized that it had already stopped. So I've got some stuff already written down that I'm not going to rewrite, because I'm lazy. Organic chemistry. Organic organism, organs associated with life. For a long time, people thought that what happened in living things was not chemistry. It was too magical and complicated. But eventually, as they learned more and more about chemistry and they got more and more curious, they went, well, let's figure out, what's a plant made up of? What's an animal made up of? And the made up of stuff is the easiest way to determine that is to dry it all out, burn it all up, analyze what's left. And when you do that, you get a lot of carbon. So organic chemistry, for a while, the chemistry of life was chemistry of carbon compounds. But carbon dioxide and carbonate rocks, there's all sorts of carbon compounds that they didn't associate with living things. We find out now they are sort of associated with living things anyway. So they went, well, what else is in here that makes it special? And the first thing that they could analyze out of what was coming out of the remnants was carbon and hydrogen. And carbon and hydrogen then became the basis. Carbon and hydrogen compounds, carbon-hydrogen compounds became the basis of organic chemistry and kind of sat there. They should have added oxygen later. They probably should have added nitrogen to HONC, even though nitrogen isn't in all important biological molecules. But carbon and hydrogen and oxygen definitely are. In fact, let me just move this over, which is what I should have done in the first place. This is what we're going to do in the next chapter, the various important biological molecules. And got a lot of carbons in them. Got hydrogens in them, also has oxygen. Carbon, hydrogen, and oxygen. Carbon, hydrogen, and oxygen, and nitrogen. There's nitrogen in here also, but there's also phosphorus, which we'll talk about later. But HONC really should be the basis of organic chemistry, and it is in reality. It just isn't by the definition. Now I'm kind of doing a PowerPoint. The simplest hydrocarbons, carbon and hydrogen molecules, are 1 carbon, 2 carbon, 3 carbons. MEP, methane, ethane, propane. And you can see when there's 1 carbon, all 4 bonds, carbon is a 4-bond molecule, or atom, remember, all 4 have hydrogens on them. Now you can put other things on there. Methanol has an alcohol group on one of the bonds, an O with an H attached to the O. We'll talk about that toward the end of the chapter. Ethanol has an alcohol group. But the plain ethane is 2 carbons and then all hydrogens. So it can't be CH8 because there's a bond between the 2 carbons that takes away 2 potential hydrogens. So there's 3 hydrogens and 3 hydrogens, so C2H. I don't know why I wrote that down. H6. I said H6 when I did it the first time. When you string 3 carbons together, the middle carbon loses 2 bonds because it's being held by a carbon on both sides. So it only has 2 bonds available for hydrogens. So you've got 3 and 3 and 2, so it's C3H8. Now these molecules, you can put a lot of stuff on the various bonds. You can put an alcohol group, you could put 2 alcohol groups. You could put an alcohol group here or an alcohol group there, or an alcohol group at this end and one of them up here at the top. And the bonds do actually hold themselves together in certain organizations. When you've got all the same atoms, but they're not all in the same place on a molecule, you have isomers. Same chemical formula, but the molecules are not the same because where they're holding the various components is on different atoms. an alcohol and an alcohol here. The same molecular formula, an alcohol and an alcohol here. So three-dimensionally, there's a difference between alcohols and alcohols. Those are stereoisomers, where the bond connections and the bond angles are different, again, between molecules that, from a pure molecular formula, are the same. Now, if you put two molecules next to one another and they match up, but they don't match up, they're mirror images of one another. Now you're dealing with chiral molecules. Now we're getting into the language. Remember, this is like a foreign language. There are terms that people expect you to understand. And if you don't understand these basics, it makes a lot of papers that you're going to read really confusing. And I will admit that for some of these terms, I have trouble pulling them back out of my memory. And it is annoying to read and go, okay, I got to look this, what this particular type of conformation is, cis and trans and things that we apply to sexuality also apply to molecules. But I realize I really need, if I were doing this all the time, I definitely would need to knock these things into my head. So I didn't. It is annoying to have to look up basic terms because your brain doesn't want to hold on to them. So get your brain to hold on to them. Now we're going to hit some basic terms. Large molecules have bonds at all sorts of different angles. They curl back around themselves. They very often present an outer face of electron clouds. The molecule has a shape. It has a conformation. And a change in conformation changes what the molecule can and can't do. A couple of examples of that. You make digestive enzymes in your pancreas, but you would prefer them not to digest your pancreas while they're there. So they are produced in one conformation, one shape that isn't digestive in function. It's a transport shape. Then they're transported to the space, the lumen inside your intestine, which is protected from them by mucus. They can't get to digest the lining through the mucus. And then the conformation is changed into an active digestive molecule. So there's a lot of reasons why shapes change. If you are using an enzyme and you're going to use it later, but you don't want to use it now, one of the ways to shut it off is to change the shape of the enzyme so it's not functional, and in a way where you can change it back later. There's a very weird sort of disease-causing molecule called the Prion that its chemical formula and the sequence of the molecule itself, of the protein, matches a protein that's functional in your cells, but they have different shapes. The weird thing that a prion can do is it can grab one of those functional molecules and change it, and when it releases it, the functional molecule is changed into the shape of the prion, and it can go on to grab more functional molecules and turn them into prions, which gradually depletes all your functional molecules, which messes up with your function. That's why it causes diseases. But that is a conformational change. It doesn't change the chemistry of the string. It doesn't change the order of the string. It just rearranges the basic hydrogen bonding and other sorts of things. Now, in these larger molecules, if you have double bonds between your carbons, then each carbon in the double bonds could have an additional hydrogen on it, right? If there was a single bond, this carbon would have a bond available and this carbon would have a bond available for hydrogens. If there's only single bonds in the molecule, it's holding all the atoms that it can hold, it's saturated. And this doesn't always apply to the whole molecule, sometimes it just applies to a component of the molecule, it happens in lipids and such. where there's a string of fatty acids, and if the fatty acids have all single bonds, they're saturated. If it has double bonds, then it could hold more hydrogens, and it's unsaturated. You hear about saturated and unsaturated fats. Polyunsaturated, a couple of double bonds, poly more than one. And it changes the chemistry of the molecule. Actually changes the three-dimensionality of the molecule. You'll notice on some of our molecules later that like phospholipids have a kink in the fatty acid. That's because the double bond redirects the direction of the string. Now, there's more things in biological molecules than just hydrogen, oxygen, nitrogen, and carbon. Some of the more important players are phosphorus. There was phosphorus in the DNA and the nucleic acids. There's phosphorus in ATP. It shows up in a lot of places. It's important. Sulfur. Sulfur is on the same column of the periodic table as oxygen. It's column 6. It has 2 spots, right? It's 6 shy of filling to 8. Has 2 bond spots. And so like oxygen, it very commonly forms a bridge between an atom and another atom. But being farther down on the periodic table, it's a bigger atom. So it forms more distant bridges. So it can hold a molecule together in ways that oxygen is not capable of doing. And there's other atoms that you find here and there sprinkled amongst the various molecules. And minerals. A lot of times you'll find minerals sometimes integrated into a larger biological molecule, and sometimes kind of stuck there, kind of held in place, but not really covalently bonded in there. What they call the heme group, the iron group in hemoglobin, is held that way. But there's a lot of things like chlorophyll doesn't work without magnesium in it, and hemoglobin doesn't work without iron in it. There's a lot of compounds that hold copper, all sorts of things. So they can be important players, and a bunch of different ones. Okay, I think the last part of the chapter is about functional groups. I'm going to stop the video here and pick that up for the functional groups.